Hello, and thank you for joining us today for part 3 of our webinar series on characterization of crystalline structure in foods and pharmaceuticals. You'll find parts 1 and 2 in the TA Instruments webinar library. Part 1 dealt with the basics of using DSC to measure crystallinity. Part 2 was a relatively new term or topic for many that dealt with what is called apparent melting. And as the name implies, it's something that it's an endothermic peak that looks just like a normal melting peak, but as we discussed in webinar 2, that endothermic peak is really the result of a chemical process taking place in the material. And that was a new subject for many. The subject for today is also going to be new to many, and that subject is how we can use apparent melting to detect and analyze drug-excipient incompatibility. Well, that's a big name and a long term, but really what we're dealing with is trying to detect interaction between two materials when they're mixed. So even though drug-excipient incompatibility is a pharmaceutical term, anytime we mix materials together, there's the potential that we'll have that interaction. And so in the food industry, it's going to be just as important because typically most foods have multiple components to them. The agenda for today is shown here. Since we have some relatively new terms and topics, I'm going to do a little bit of background. And that background is going to include a discussion on quasi-isothermal modulated DSC, a technique that's been around for probably 25 years, but it's not a common technique, so I'll just do a little refresher on that. Following that, we're going to talk about what was the objective, what we were trying to achieve with this work. I'm going to illustrate I'm going to propose the thermal analysis method that we're going to use for this. I'm going to illustrate that method and then do a summary and then finally try to answer any questions that you might have. Before I begin, it's necessary for me to thank a number of folks who have allowed me to use their work in this presentation. From the University of Illinois in Urbana-Champaign, my colleagues include Professor Shelley Schmidt, Dr. Ju-Wan Lee, and Dr. Lydia Liu. Here at TA, most recently at the NATIS conference, Jarita Sanchez presented a paper which included work on drug-excipient incompatibility, and I'll be using her work as well. So thanks to my colleagues for allowing me to use their work. All right, so let's start with the background. Again, drug-excipient incompatibility is probably a new term to many, especially if you're not in the pharmaceutical industry. But as the name implies, we have a drug, which can be called the active pharmaceutical ingredient, API. And in order to deliver that to the patient, we're going to put it into a tablet form, a capsule form, whatever. But whenever we do that, especially in the tablet form, there may be another half dozen or 10 ingredients that are required in order to achieve the desired properties of that pill. So what we're going to do is take our drug and we're going to start blending it and mixing it with numerous other ingredients. Typically, the drug is maybe no more than 10 or 20% of the pill's weight. So we've got lots of other things there. The concern is that over time, that drug is going to chemically interact with one or more of those other excipients or components, and that's going to affect the efficacy of the drug. If there is a chemical interaction between the drug and one of those other materials, then it can affect what's listed there: chemistry, physical properties, stability. And in reality, when you have a chemical reaction between two materials, all of those properties are affected. And so in the pharmaceutical industry, you obviously need to know if that's going to happen because the materials are going to be stored for a long time and matter of years. And so you don't want that type of degradation taking place in your materials. So the focus today is drug-excipient incompatibility, but again, anytime we mix materials together, there's the potential for interaction. If you joined us for webinar number 2, you have a pretty good understanding now of what apparent melting is. And again, apparent melting looks like melting. It's an endothermic peak in our DSC data that results in the loss of crystalline structure. But it's not the melting process that's driving that loss of crystalline structure. With apparent melting, we have a chemical process that is causing the loss of the crystalline structure. And in webinar number 2, we provided numerous examples of processes. And they included decomposition, chemical reactions, dissociation of salts, desolvation of hydrates, denaturation of biological materials, proteins. And today we're going to talk about another component to that list of chemical reactions, and that's the drug-excipient incompatibility, where we have a chemical reaction. Now, the reason that it's so important to understand apparent melting and its application to drug-excipient incompatibility is that with apparent melting, regardless of any one of those processes listed there, there is always a chemical change. And remember, we can never allow a chemical change to take place in our drug, otherwise everything else changes. So apparent melting is going to point out anytime we have a chemical change, and that's why it's such an important part of this application. I mentioned quasi-isothermal modulated DSC is not a very common technique. It is used, but in this case, you're going to see that it has a fundamental role to play in this application. Now, let's just explain, or let me explain what quasi-isothermal modulated DSC is. It's a long name for a simple experiment. Basically, what you're going to do is you are going to pick an isothermal temperature to do your experiment. And I'm going to show you how you can zero in on the right isothermal temperature for your MDSC experiment. So once you pick your isothermal temperature, we want to be able to measure heat capacity changes at that temperature. And the next slide will explain why heat capacity is such an important property to measure. In order to measure heat capacity at some isothermal temperature with modulated DSC, what we're going to do is to apply a small temperature modulation on top of that isothermal temperature. And one of these days I'll probably do a webinar on modulated DSC, but for now let me tell you what the best conditions are for this kind of experiment. If we're trying to measure changes in heat capacity with time, I'd strongly recommend that you apply a temperature modulation of plus or minus 1 degree every 100 seconds, all right? Now, using those conditions, we would end up creating a temperature profile that's illustrated here in this particular slide. If we go to the green axis on the left-hand side, the green y-axis, that's a plot of temperature, and that's the measured average temperature during our experiment. On the right axis, the blue axis, we have modulated temperature, okay? And if we look at the blue curve, what we see is that that blue curve is changing sinusoidally. If we look at the axis on the right, we can see that that sinusoidal temperature change is occurring between 139 and 141. So that's our plus or minus 1 degree C modulation amplitude. Since it's a sine wave, we have to define the time period, the frequency of that sine wave. And again, the recommended condition here is 100 seconds. Believe me, you can use many other conditions, but we're not going to get into how we select conditions today. That would be something that would be done during a modulated DSC webinar. Hopefully I can do that sometime in the coming year. So that's the temperature basis of our MDSC isothermal experiment. Now let's talk about why we want to measure heat capacity. We're dealing with a crystalline drug, or we're dealing with a crystalline excipient. One of the two materials needs to be crystalline, the drug or the excipient, or in order to use this methodology. Okay? And so if we start with a crystalline drug and there's chemical interaction, what happens is we start to break bonds, the crystalline structure becomes amorphous. And there's a big difference in the heat capacity between crystalline structure and amorphous structure. And generally, the structure changes by as much, or the heat capacity changes by as much as 50% on average. So we have a big change in heat capacity that is easily followed using the modulated DSC isothermal technique. Now, in the case of why we can do that, heat capacity is not a rate property. It's an absolute thermodynamic property. If you're using DSC, you know that the faster you heat, the larger the heat flow signal. And if we look at the units for the heat flow, it's milliwatts or watts per gram, where a watt is a joule per second. So in a DSC, we measure heat flow rate. And the faster we heat, the larger that signal becomes. But that's not true with heat capacity. Heat capacity has absolute values as a function of temperature. So it's an absolute thermodynamic property, and it doesn't have a rate function. The units of joules per gram per degree C has no time dependence to it. So it doesn't matter how fast we heat in terms of following heat capacity with the modulated DSC. And you'll see why in the next slide, why we can do this, okay? Now, in the case of standard DSC, you don't measure heat capacity. What you measure is heat flow rate. And so we use that heat flow rate to calculate heat capacity using the little equation that's listed there. Heat capacity is equal to heat flow rate divided by heating rate. But if the heating rate is 0, obviously you cannot make a measurement. With modulated DSC, we have two simultaneous heating rates being applied. One is the average heating rate, which we can set as 0, isothermal. The other is the modulated heating rate, which we control by changing the amplitude and the modulation period. And we'll see that in the very next slide. So with modulated DSC, reversing heat capacity is equal to the amplitude of the modulated heat flow signal, that's a measured signal, divided by the amplitude of the modulated heating rate signal, again, another measured signal, times the calibration constant for reversing heat capacity. Now, it is not necessary to actually calibrate for heat capacity for this application. The reason is we're going to be looking for the rate of change, and it doesn't matter whether the unit is calibrated for heat capacity or not. Typically, that calibration constant will be about 1.05 to about 1.1 for most of the DSCs that you'll be using. So it's not necessary to do the calibration. If you want to do the calibration, get absolute heat capacity units, then you're going to calibrate probably using something like Sapphire. So that's how we're going to, that's the equation we're going to use to follow heat capacity. And let's take a look at what those signals would look like. If we look at the inner axis on the y-axis, the left-hand red axis, I've set that up just to plot temperature. So we can follow the measured average temperature during the experiment, which was selected in this case to be 140 degrees. What we saw in the previous slide was that the temperature is modulating plus or minus 1 degree every 100 seconds around that average 140 degrees. And so if we look at the inner axis for the brown or maroon curve, what we see is that that curve is centered near 0 in value, but the lowest heating rate is a -3, which means there's some cooling, and a +3, which means there's some heating. So, and you would expect that if the average heating rate is 0, we're going to be oscillating over that range between a -3 and a +3. So the heating rate is changing sinusoidally. Because the heating rate is changing sinusoidally, the heat flow signal measured heat flow signal is also changing sinusoidally. And so in order to get the reversing heat capacity signal, which is shown here in green for sucrose at 140 degrees, all we need to do is take the ratio of those two measured signals. So it'd be the amplitude of the modulated heat flow signal divided by the amplitude of the modulated heating rate signal. So it's a really simple experiment, and we can follow changes in heat capacity as a function of time at some selected temperature. Now, in terms of the objective of this presentation, of the research that we did in trying to develop the application, what we're trying to do is to come up with a technique that provides rapid screening for drug-excipient incompatibility. Why rapid screening? Because if we look at what is currently done in most pharmaceutical companies is that in order to detect drug-excipient incompatibility, they make up mixtures of the drug and the excipient or excipients, put them in an oven, typically at a range of temperatures, and then periodically over time, over months, do chemical analysis to determine if something is changing or not. So we're talking about tying up ovens and analytical equipment for months at a time to detect drug-excipient incompatibility using current technology. The proposed method can be done in 1 to 2 days, and that 1 to 2 days would be to run the drug, to run the excipient, and I'll be demonstrating this or illustrating this, but to run the drug, run the excipient, to determine whether there's some kind of interaction occurring. As you'll also see, once we detect this interaction, we might be interested in the rate of that reaction, which helps us project onto lower temperatures and longer times, which would be important in terms of knowing what the possible shelf life of this material might be. But it also allows us to do chemical analysis using any analytical technique that you want. And you'll see how that happens here as we go through this presentation. So the proposed method was to give us a rapid screening tool, but it doesn't replace the need to ultimately do that oven aging over a period of time, because this is going to hopefully weed out the bad materials. But once you select what you think are good materials, you still want to be sure there's no interaction. So it's that likely formulation that you're going to be putting in the oven and doing your longer term testing on. Okay, the proposed method that we're using here involves 3 steps to it. One is thermogravimetric analysis, TGA. Excuse me. And in the next slide, I'm going to talk about what we're looking for with the TGA. We're also going to do DSC on the drug and the excipient at multiple heating rates. And you know why we're using multiple heating rates based on apparent melting, and that was completely or thoroughly discussed in the previous webinar, but I'll just give you some examples here today just to refresh your memory. The third step is probably the most important step, and that's where we're going to mix the drug with the potential excipient, and we're going to do some DSC tests at a low heating rate to determine whether or not there really is interaction occurring. So that's going to be the critical step to determine whether there is interaction or no interaction. Now, in case of the TGA, excuse me again, in case of the TGA, I'm not going to be using any data here today, and that's strictly because of the time limitations of the webinar. But there is a new applications note out, TA404, where we do present the TGA data. So if you're interested in this, get that app note in the TA library, and you'll be able to see the TGA work that we did. But there's a quick summary that we can give you. Basically, we're going to use the TGA as a way of verifying composition. And specifically, we're going to be looking for free water, because that can affect as a plasticizer, reaction, et cetera, hydrolysis. We're going to be looking for bound water or bound solvent, because if we have dehydration, desolvation, we can create an amorphous material that is more reactive, and if we're freeing water, then that of course can contribute to the reaction as well. So we're going to be looking for hydrates. Before you put your material into a DSC, you want to know where the bulk mass loss is. All of these experiments should be done in hermetic pans, but once you start heating this material and start decomposing this material, once you have a 5 to 10% weight loss, you start to create a lot of pressure in that hermetic pan. And so the pan can leak or burst and you'll contaminate your DSC. So doing the TGA first tells you at what temperature you have that bulk mass loss. And that is probably going to be the limit temperature that you'll use then for your DSC experiments and modulated DSC experiments. And lastly, the TGA is really helpful. Once you know what the components are, you can use that information to help with your interpretation of the DSC data. After we have done the TGA work, then we go to DSC at multiple heating rates. And the purpose of the multiple heating rates is to determine whether we're looking at real melting of these materials or apparent melting. If it's apparent melting, then there's a couple of issues, and they're listed here. If we have apparent melting and we start to measure the temperature of the melting peak, temperature or the heat of fusion of that melting peak, what we find is that if we have apparent melting, there is no constant value. The faster we heat with apparent melting, the higher in temperature that endotherm occurs, and the larger the peak becomes in area. Not a big change in area, but it does become larger, and that's due to the temperature dependence of the heat of fusion, which was discussed in webinar number 2. Now, if you have apparent melting, you may be interested in knowing something about the rate of that process, how it extrapolates to lower temperature and longer times. And so the very first step would be to take our data, and I'm going to illustrate this, and do an Arrhenius plot. And there's several ways of doing that Arrhenius plot, and we'll show you those ways. In addition, if there's a chemical reaction taking place between our drug and excipient, then we might want to know what that reaction is, because if it's an acid-base type reaction, then we know that if the excipient is basic, then we might be concerned about other materials that are basic as well. And so once we know there's apparent melting, we can use DSC and MDSC to look at the kinetics of the reaction as well as the chemistry of that reaction. And that'll be illustrated. Some of this is a slide that you saw in webinar number 2, and it's on sucrose that is made from sugar cane. And this is an expanded view of the baseline for the sucrose sample. What was important here was that if we started our experiment, say, at 25 degrees C per minute, and I've got to thank my colleagues at the University of Illinois who this is some of their work, but if you started at 25, that result would look like the green curve that you see there. And so you have certain temperatures associated with this minor endotherm and the larger endotherm. The blue curve was done at 10 degrees per minute, the red one at 5, and the maroon one was done at 1. So what you clearly see is that the higher the heating rate, the higher the temperature of these transitions. And that's because the loss of crystalline structure here is being caused by hydrolysis, which I'll briefly mention in the next slide. But here's the biggest benefit, I think, of this slide. Once you have the data for your 1 degree per minute experiment, whether that be the material, the drug by itself, the excipient by itself, or the mixture, once you have that 1 degree per minute data, if there's apparent melting, the rule of thumb is, and this is my rule of thumb, it's a guideline, pick your isothermal modulated DSC temperatures in the range between 5 and 20 degrees below that 1 degree per minute data. So that's going to put you down where that vertical 120 line is shown. So we're going to now look at how this crystalline structure is lost due to this chemical process at a temperature that is below any of the obvious transition temperatures that we see in your data. If we take that data, I said we can do a kinetic analysis of it, and that starts with the Arrhenius plot. So if you look at the y-axis here, it's log heating rate, and those were values of 1, 5, 10, 25 degrees C per minute. But we're looking at log heating rate on our y-axis versus 1 over temperature for the onset of the transition temperature. If we make that plot, we get the values that are shown here. It creates a very nice straight line, which tells us that there's a single process that is causing this to occur. And in the numerous papers published by my colleagues at the University of Illinois, they clearly show that this process is hydrolysis, okay? And that was discussed in more detail in webinar number 2, as well as in many of the papers that they've published. Okay, so we said that once we know there's apparent melting occurring, we might want to do further kinetic analysis, we might want to do chemical analysis. And so the first thing we do before we go to those isothermal modulated DSC experiments is determine the best temperature to do those analyses as a function of time in order to get kinetics of versus temperature as well. All right, so here is that same sample of sucrose, and it's sigma sucrose, so it's an analytical grade, highly pure sucrose. We're looking at reversing heat capacity on our y-axis versus time. Now, in this case, this experiment was done by one of the students in the laboratory, and when you're a PhD student, you're willing to stay in the laboratory for maybe 50 hours. But this is probably a longer experiment than was necessary, especially since we know that it doesn't matter how fast this reaction goes. The kinetics data showed that it's one process causing this. But, and my guideline would be to pick your temperature when you have about 8 hours for a complete loss of crystalline structure. And I'll show you that in a later slide. But when we look at this figure, we can see that at 120 degrees, which is nearly about 55 degrees below the often reported melting point sometimes in the literature, what we see is that we immediately have increasing heat capacity with time. So even at 120 degrees, there is a chemical reaction taking place, which turns out to be hydrolysis, and we have conversion of our crystalline structure to amorphous. Amorphous structure has a higher heat capacity. So as we convert from crystalline to amorphous, we can follow that rate by following the increasing heat capacity. In this case, we knew how much change in heat capacity was required for 100% conversion, and that was about 0.68 joules per gram, and that's about the value that we got here. So we believe that at 50 hours or so, you have complete conversion of the crystalline material to amorphous. Now, this is using the recommended conditions, which are shown there. We've got about 10 milligrams. This was done at 120 degrees, 100-second period, plus or minus 1 degree C amplitude. So that's an example of using our quasi-isothermal modulated DSC to look at this reaction process. Now, once we have that profile, if we're interested in understanding the chemistry of this reaction, there's a very neat, simple process that you can use. Once you know that profile and you select your temperature for looking at the chemical change, what you can do is prepare your samples and place them into an oven at that temperature. Okay? Now, you know what % conversion takes place versus time at that temperature. And so what you can do is, as they did here, and you can see the values indicated, what you can do then is pull samples at that particular point in time, take them, put them in the refrigerator to stop the reaction. So when you have all of your samples built up from different time periods, then you can go ahead and do your chemical analysis. And the nice thing about this process, or this method, is that if you're most comfortable with HPLC, which they used to identify the decomposition products here, then use HPLC. But if you want to use IR, if you want to use GC, you want to use mass spec, you have the samples now in your vial that had been subjected to this temperature profile. You can go ahead and use any analytical technique you want to do the chemistry. And what they found with sucrose is that the decomposition products were fructose, glucose, and 5-HMF, which I think is hydroxymethylfuran, okay? So you can follow the decomposition process by following the decomposition products using the quasi-isothermal modulated DSC data, shown as that gray background, as a guide to where you are in the reaction. In terms of illustrating drug-excipient incompatibility, we're going to use probably the classic example of this issue between drug and a potential excipient. And there are numerous papers in the literature on it, and here we're looking at one of the websites. I think this was called Pharma Treasures. And what they did was they had a little blog on what happens when you mix aspirin, acetylsalicylic acid, with mag stearate, okay? They're not compatible. They're known to react. So how does our method look when we start working on a system that we know is not compatible? First thing we do is run both the drug and the excipient at the multiple heating rates, the 1, 5, 20. And with the aspirin, what you see is that when you heat it 20 degrees per minute, you get an onset temperature of about 141. When you drop the heating rate to 5, the onset temperature moved down to 138. At 1 degree per minute, the onset temperature was 131. So clearly, there's no constant temperature for the conversion of crystalline structure to amorphous. And that's because there's a kinetic process driving this, and that's apparent melting. Okay? So anytime you have a shift like that, that's apparent melting. We would then look at the excipient, so we know what that looks like by itself. And in this case, the magnesium stearate had both some hydrated crystals and anhydrous crystals in it. It was right on the label that both forms were there. And it was kind of an interesting sample because what we see is as we do these three heating rates, 1, 5, and 20, we get 2 peaks for each heating rate. And using the TGA data, we confirm that the first endotherm is the result of the dehydration step. Okay, that's where we're breaking off the water molecule. Since there's a chemical change taking place there, it's not surprising that when we change our heating rate 1, 5, and 20, that first peak shifts with heating rate because there's a chemical process behind that conversion from crystalline to amorphous. But the anhydrous crystals went through true thermodynamic melting. So we look at all 3 heating rates, that high temperature peak doesn't shift with heating rate, and that tells us that that's a real melting process that's taking place there. So now we've run the drug, we've run the excipient, we know what they look like individually. And our third step then is once we have that individual data, we're going to make up our 50-50 mixture and do a slow heating rate experiment. Now, in terms of that 50-50 mixture, completely arbitrary. I find that this works probably as good or better than anything in terms of what mixture ratio of mixtures to use. Okay. Remember, our goal here is to simply detect whether there's a reaction taking place. So we're going to try and maximize the concentration of both the drug and the excipient, and we do that by using a 50-50 mixture. But if you want to make it on a molar basis, you can. If you want to look at different concentrations of excipient, you can. It's completely up to you because you're just going to mix them up and put them in a hermetic pan. Now we're going to use the 1 degree per minute data, and I'll illustrate this, but you're going to use it because, remember, you got 2 materials that are potentially going to interact. So what you want to do is heat slow to give them time to react if they're going to react. Now what we're looking for in this mixture data is we know where the individual materials have their transitions. We want to see what happens when we mix them together. Do the transitions remain at the same temperature, or do they move? And if they move to a lower temperature, that means it took less energy to break the crystalline structure within these materials, which means that there's some kind of chemical interaction taking place. If we see this interaction, that's where we can then decide if we want to do chemical analysis, as we saw with the sucrose, or do kinetic analysis. All right, so here's that step 3. We had our aspirin, acetylsalicylic acid, which we ran at 1, that's the green curve. We have the mag stearate from 1 degree C per minute, and that's our blue curve. Mix them together, and I did this experiment myself, so when when you take two crystalline white powders and you mix them, it's easy to see if there's any fizzing or reaction taking place, okay? And there wasn't. I just had two nice white powders, easily mixed, that I could use for this experiment. So I know there was no reaction that took place prior to heating it in the DSC. But look what happens. We end up with the data that's shown in red. So simply by making a mixture of the blue sample and the green sample, the magstearate and the aspirin, now what happens is we end up with a peak at lower temperature. And that peak is apparent melting. As soon as we have the chemical interaction taking place between those two materials, it breaks the breakage of bonds causes a loss of crystalline structure. So that's the cause of that endotherm that we see in the data. And what we see then after that is that we have some exothermic reactions taking place. But clearly, something is happening to the mixture of the material. Now, if we look at that red curve, we can see that the extrapolated onset temperature is about 68 degrees. And using the rule of thumb, that would say we want to go down to maybe 48 degrees to as much as 65 degrees or so to start doing an isothermal experiment. So that's the rule of thumb. Take that onset from 1 degree per minute and pick temperatures in the 5 to 20 degree range below that to do your isothermal modulated DSC experiments. Now this is work from one of my colleagues, Jaritza here at TA, where she selected isothermal temperatures of 40, shown in green, 50 in blue, and 60 shown in red. So what we're looking at is on our y-axis, the reversing heat capacity versus time. And so since we saw that the extrapolated onset temperature was at 68, it's probably no surprise that at 60 degrees shown in red, that curve has a lot of steep slope to it. The reaction goes very quickly at 60. Once we've eliminated all the crystalline structure, the value for heat capacity will plateau until you get significant decomposition, then you might see a change one way or another. At 50 degrees, the reaction went a whole lot slower, okay? And that gives us the slope that we see there. Notice that at about 600 minutes, a little more than 8 hours or so, that the value levels off, okay? That would actually be a good temperature for doing your chemical analysis because you have a window then of about 6-8 hours that you can be pulling samples, putting them in the oven, doing your chemical tests the very next day. At 40 degrees, the reaction is really going very slowly, but you can still see the slope. Now, the software will measure the slope for you. You just pick your limits, and those limits are shown on there. You pick your limits, the software will measure that slope. Now, we're going to take that measured slope to get our kinetic information. So on the left-hand axis here, we're plotting log of reaction rate. In the previous Arrhenius plot, we plotted log heating rate. Here from the MDSC isothermal data, we're plotting log reaction rate, again, versus 1 over temperature. And so for our three temperatures, 40, 50, and 60, we again see that there's a pretty good straight line indicating that there is a single process that is driving that reaction. Now, hopefully, not all of your experiments will show chemical interaction because you're looking for materials that don't interact. We had a paper where it stated that there may be some chemical interaction between a particular drug and lactose. So what we did was some experiments to find out whether that was true or not. So I can't identify the drug. It's a commercial prescription drug, but it had two active drugs in it, and one of those active drugs was a salt. Now, this is the tablet that had other components in it ground up. So we're looking at that ground up tablet. The first thing we do is run the tablet by itself. So the tablet by itself, ground up, at 20 degrees per minute is shown here in green. So we clearly see that there are two endothermic peaks. We don't know which is which here in terms of the identity of the material causing those peaks, but we have two peaks. If we drop our rate to 5 degrees per minute, which is our blue curve, we again see two distinct peaks. Now, notice that there's a little better resolution between those two peaks at 5 as compared to 20, and that's very normal. The slower you heat, usually the better the resolution of overlapping transitions. Our third heating rate was the slowest heating rate on just the tablet itself, and that's shown in red, and that was at 1 degree C per minute. And at 1 degree per minute, again, we're getting very good resolution, maybe to the point where we can see that there's actually a minor third component there in the middle between the two major peaks. So we now know what the drug does as a function of heating rate. When we looked at the lactose, it melts above 200 degrees, so we really didn't care what was happening up there with the lactose, although I think we did the experiment at multiple heating rates, and it too goes through apparent melting, but it's way up there in temperature. So what we did was take the ground tablet and take a 50-50 weight mixture of the ground tablet with lactose. Now the question is, do one of those excipients, or one of those drugs interact with lactose? And that's our black curve here. Now notice that with the black curve, which is the 50-50 mixture, that the onset temperature of that lowest peak really doesn't change at all. In fact, the overall shape of the curve doesn't really change at all from the 1 degree per minute data. We start to lose that minor transition in the middle, but the fact is that 50% of this sample is now lactose that has no transition there, so it's not surprising that we might not actually see that minor transition. What we're looking for is how does the 1 degree per minute data in the mixture compare to the 1 degree per minute data from the individual compounds? And what we see here is that there is no change. That tells us that there's likely no interaction between lactose and either one of these active ingredients. Okay, I'm going to wrap up here, and then we'll try and take your questions. So in summary, it's really important to the pharmaceutical industry or to other industries that are mixing materials together to determine if there's any interaction that might be taking place between ingredients in this formulation. And regardless of your industry, you know, there's got to be some shelf life that you need. You can't exactly control temperature. Maybe in the pharmaceutical industry you could, but in general you can't control that final temperature. It's going to typically be cold sometimes and hot another time. So you want to know, is there the potential for interaction between my ingredients? Although crystalline material is the preferred form for drugs because it is more stable, the fact is that crystalline materials can react in the solid state. We've showed numerous examples in the webinar on apparent melting. Okay, so just because it's crystalline doesn't mean it's necessarily stable once you mix materials together. Typical test time for the industry in terms of detecting drug-excipient incompatibility is many months, and you're going to do a lot of analytical work during those months to test for chemical changes. The technique here that we based on apparent melting allows us to greatly reduce this time to determine if there's drug-excipient interaction occurring. The proposed method is based on TGA, DSC, and MDSC, as you saw. And not only is it relatively fast, but it gives us the potential for doing the chemical analysis, as we saw. So if you want to understand the reaction mechanism so that you can avoid similar excipients in the future, you can do that. It provides kinetic analysis, and kinetic analysis is derived from the temperature range that we used. But once you see that you have a linear plot, you can certainly try to extrapolate to lower temperatures, longer times, to try and get an estimation of shelf life. Now, the fact is there's no way that we could get into kinetics beyond the activation energy pre-exponential factor today. But if you're interested in now taking the information that you obtain here and doing complete kinetic analysis on it, Dr. Roger Blaine has published, has created 2 webinars here on the TA Instruments Library that go into detailed discussion of kinetics. And Roger is very active in ASTM, and he'll probably recommend in his talks that you obtain the book from ASTM, which talks about all the different kinetic methods. So we can't get into the kinetics today, but the first step of getting into kinetics is information that we can derive here. Lastly, I believe that you'll find this method very easy to use, very easy to interpret. There's not any difficult determinations here. Does it shift or doesn't it? Isothermally, do we see a rate or don't we see a rate? So it's pretty simple to do the analysis of your results. Now, had to do this pretty quickly today. Didn't show you the TGA data, but if you're interested in this subject, then I would suggest that you go to the TA library and look up TA 404, which will have much of this material plus additional material in it. So thank you for joining us today. And now what I'm going to do is try and take a look at the questions that you've been submitting, and hopefully I can answer them. Thanks again.
Okay, I hope everybody can hear me. I'd just like to thank everyone for joining us today. I've got about a half dozen questions here, and I see more coming in, so let's get into some of these. And the question is, what could be the effect of different metal type of pan on CP of drug? There aren't many options that you have in terms of pan types. Generally, you're going to be using aluminum because it can be hermetically sealed, or you might go to stainless steel, which it can be hermetically sealed, but I would not recommend the stainless steel simply because those pans weigh typically 250 milligrams or higher. Although there are other types of pans, whether they be platinum or copper, those, to the best of my knowledge, those pans cannot be hermetically sealed, and so you would not want to use other pan types. So let's assume you could come up with copper pans, say, that could be hermetically sealed, there would be no benefit to using copper over aluminum unless the materials you have might react with aluminum. So the assumption is when you pick a pan type that the assumption is that there's not going to be any chemical interaction between the sample and the pan. So that's one assumption. A point that I think I made in the presentation was that you need to be using hermetically sealed pans. That's really critical, and that's because as you heat the material up, if there was any volatile component and you were running in a non-sealed pan, then you can start to eliminate some of the water or solvent that might be in there. And that water and solvent can act as a plasticizer and affect the reaction. So generally you want to be using aluminum pans and specifically hermetic aluminum pans. Another question is, can the change in heat capacity be used to quantify the amorphous content? And the answer is it absolutely can. If you're working with either the drug or the excipient, then you can actually determine what the change in heat capacity is in going from crystalline to amorphous by using just standard heat capacity measurements, whether they be modulated DSC or one of the other approaches. But when you, in terms of drug-excipient incompatibility, now you're dealing with a mixture of materials, and so it might get to be a little complicated to try and quantify the conversion from the change in heat capacity. The most important point here is that if there is interaction between a crystalline material and anything else, that the heat capacity will be increasing with time. And so it probably doesn't matter if you can quantify the change as much as determine whether there is a change or is not a change. Next question is, what about applying this method to a mixture of amorphous drugs with a crystalline excipient? And the answer to that question is that only one of the materials needs to be crystalline, whether that be the drug or the excipient. It really doesn't matter. And the reason that one of them must be crystalline is that you're looking for the increase in heat capacity with time. And so as crystalline material becomes amorphous, then the heat capacity will be increasing. Of course, the other thing is that if you're looking at the mixture as you're doing the 1 degree per minute ramp, all that you really care is, did the mixture create a lower melting temperature than what you got in the pure material? So, in short, one of the components that you're looking for in this method must be crystalline. Let's see. I am wondering if you are rerunning the same sample of each at different heating rates, or are you using fresh samples each time? The answer there is you must use fresh samples each time. You would not want to create a different thermal history for each of those runs. So you're always going to be using fresh samples for each of those rates. Question, how does it differ from sample kept at 4075 RH versus the technique? We won't be seeing the effect of moisture. Well, so the question really is a question of do you have a sample that might be conditioned at different temperatures with different RHs? And certainly what you can do is you can precondition any of the materials before you make your mixture, and the samples that are conditioned at higher RH are most likely going to have a higher water content. That water content is going to act as a plasticizer if any of the materials are amorphous. The key thing is that, again, as I said before, whenever you have volatile components, you always have to make sure that you're doing your experiments in hermetic pans. Otherwise, the composition can be changing. So there's no problem with preconditioning your materials to any particular temperature in RH before you actually make the mixture of the drug and excipient to look for the incompatibility. And next question, how do you pick between the different 5 to 20 degree per minute rule for the isotherms? What makes one better than the other? And the answer there is that 5 to 20 is just a rule of thumb. It's most likely you're going to find some value in between 5 and 20 as a good isothermal temperature. Here's why it's nearly impossible to give you an exact value, and that is that what we're dealing with here is a chemical reaction, and every reaction is going to have its own activation energy, which tells you how much it shifts with time and temperature. And so what you really have to do is, work in, what you really have to do is, work in that 5 to 20 range. And you may find that you need to go inside or outside that range. But 5 to 20 has seemed to work for all the experiments I've ever done with different materials. Another question, any application in detecting interaction between a protein API and an excipient? Well, I have no experience in looking at protein drugs with excipients, but I think that the answer here is the same, and that is, if you have a crystalline material. Let's assume that the protein has some crystalline structure in it, or the excipient has some crystalline structure in it. If you have chemical interaction, then that means that you're going to be breaking bonds. And if you're breaking bonds, you're going to affect the ability of the material to retain its crystalline structure. That is, chemical reactions will cause a loss of crystalline structure. That loss of crystalline structure will cause heat capacity to increase. And so it doesn't really matter, I believe, that whether it's a protein or some other form of drug to do this experiment. But I have no direct experience in looking at proteins for chemical interaction. My study involved, another question, my study involves use of customized steel type. It is the C effect of metal with drug molecule. I'm having a hard, let's see. Oh, we got more questions coming in, but do you think I can get any information from CP, different steel type? I have no experience with looking at different types of steel. The fact is, every type of steel has a heat capacity, and so you should be able to measure heat capacity to differentiate between different steel types, but that's kind of outside the subject matter here. Another question: if a true thermodynamic melt occurs instead of apparent melting, can you still use this method? And the answer is yes. It doesn't really matter if you have your experiments on the drug by itself. You have your experiments on the excipient by itself, and now you're going to mix those two materials together. So it doesn't really matter whether the individual components are going through apparent melting or real melting in terms of the possibility that they may chemically interact with each other. So it doesn't really matter whether you have true or thermodynamic melting, a true melting or apparent melting in terms of using this method. How receptive is the FDA to this type of evaluation to justify non-performing further chemical studies? And the answer is, I have no clue what the FDA might think of this, but here's I guess, the most important part to this answer, and that is that this technique does not replace the need for long-term oven aging and analytical studies. What it does is, this technique, the purpose of this technique is to identify quickly as possible any excipient that might be reacting with our drug. So if you find that there is interaction occurring, then you want to eliminate that excipient as a possible excipient and go on to something else. Once you think you have found an excipient that is stable with your API, then you're going to subject that to the typical oven aging with different humidities and different temperatures that the FDA requires. So this is just a rapid screening tool to eliminate likely problem excipients, and then you're going to, once you find good candidates, then you go ahead and do the routine testing that the FDA requires. Okay, we got to take where questions continue to come in, so I can only have time for one more, but what we're going to do is to answer the remaining questions by email. So you will get an answer in the next day or so, but I've only got one more. How can I determine if an interaction is positive or negative with only DSC? If interaction is positive or negative, I guess that means whether there is interaction or isn't interaction. The DSC is just one technique to look for interaction. And the fact is, if you find interaction, you're most likely going to want to do chemical analysis. And so you're not likely going to just use DSC. It's just one of several tools that you're going to need to determine if interaction is occurring. Again, I think the DSC gives us that rapid screening technique to determine is there likely interaction or likely no interaction, and then that allows us to proceed on with other types of tests. All right, so because of time, I'm going to call it here, but again, for those of you that have not had your question answered, you'll be getting answers either directly from me or from other folks here at TA. Again, thank you very much for joining us, and I hope that you found that your time was well spent today. Take care and have a good day. Bye.